Today is the fun part, right? Today is the part where statisticians enjoy most, which is the analysis part. And so thank you for joining today. So we're going to be talking about different types of analyses, and we'll talk about where these occur during the conduct of a study and who we analyze and what we analyze. You will find out that most of the analysis tends to be in large clinical trials, a fairly simple type of analysis for the primary analysis. But there's always complications, there's always little considerations that make it a little bit more interesting. And so we'll be talking about some of these other challenges that we have in analyzing the data, that some of them we have in other types of studies as well, but there's some that are unique to clinical trials. Okay, so what do we mean when we say analysis of a clinical trial? People just throw out this term and what do they actually mean when they say, oh, analyze this clinical trial? It basically means analyzing the primary hypothesis. Remember that the clinical trial is hypothesis driven. It has usually one hypothesis, and that is what is the effect of the intervention on a particular outcome. The primary analysis is then answering the primary hypothesis of that relationship, that causal relationship. It is how it is stated in the registered protocol, and it's under the intent to treat principle. And what makes it interesting, though, is not just this testing of a simple hypothesis, but it is the fact that ethics, and again, ethics is what dictates this, we do not have what I would call the luxury of doing your analysis after you have all the study completed, all the end, you've reached the end of the study, all the data are cleaned and locked. So we do not have that. We have to do other types of analyses because of ethical considerations. So how do we specify this analysis? Where do we specify it? There's actually 3 different places and 3 documents that contain information about the statistical analysis. So when we develop the study protocol early on, before we get this, before we even actually have the first patient, we have already developed the study protocol. In the study protocol, there's all sorts of sections about describing the study, the design, etc. But there is a section on statistical design analysis considerations. They have, of course, they talk about the sample size, usually for the primary efficacy outcome. And then they specify what is going to be done for interim analysis and end of study efficacy, what is going to be done for safety. So these are the 3 things that we do: interim analysis, end of study, and we look at safety. But this is stated in the protocol, but pretty much like everything in the protocol, it's in general terms, okay? So it says how you're going to do things, but it's not very, very specific. Most trials do have a data and safety monitoring board, like we mentioned yesterday. And of course, they have a document that's called the charter. And the charter then describes what is the role, responsibility of the DSMB, but also includes specifics now on what are the methods for the interim analysis, okay? So here's where the interim analysis specifications are given. Before the end of the study, we have what's called a statistical analysis plan, or the SAP. And this is the place that has the end of the study descriptive, detailed description of what is going to be done for the efficacy, for primary, for key secondary. It talks about the safety analysis. It talks about subgroup analysis and other secondary and exploratory sensitivity analysis. So this is where we have the analysis specified. These are the sections that in these 3 documents that statisticians are primarily involved in writing and deciding. So this is where the excitement happens, right, in specifying this. So to summarize this, the study protocol has a draft statistical analysis plan for the end of the study, and sometimes also mentions what's going to happen in the interim. But prior to the first DSMB, the charter specifies the statistical analysis plan for the interim analysis. Why do I call it draft? Because the statistician will draft it, but who has the ultimate say-so as to what is done for the interim analysis is the Data and Safety Monitoring Board. So they may modify it and they will propose a final one. They may not like what we have proposed, for example. And that's where you'll have the final analysis plan. And as I said, prior to unblinding the study results to the investigators, the final statistical analysis plan is developed, and it talks about all of these issues as well. Now, if something is not specified, some analysis is not specified in the SAP, they should be called post hoc analysis. And what happens is once you start looking at the results of the study, people are now unblinded as to the results. They will think, oh, why don't we do it this way? Why don't we do it that way? And those are analyses that are after you've seen the data. And so that is now by regulatory agencies, they say, you must call those post hoc analysis. Now, journals don't keep people to it because they don't know. And sometimes they don't go through the exercise of going and looking at what you register. But the statistical analysis plan has to be registered, signed off, approved, and they may demand you to look at it. And if they look at it and they see that it's post hoc, they will force you to call it post hoc as well. So before we go into the details, statistical details of it, the next point that's important is who do we analyze? Okay, and we call those the analysis populations. We've heard the term intention to treat. Intention to treat is every subject who is randomized according to the randomized treatment assignment is included in the analysis, okay? So the point here is that you ignore anything that happened afterwards. Maybe people did not comply with the protocol. People did not comply with the interventions, did not adhere to it. Maybe your staff did not follow the protocol properly. Maybe subjects withdrew or something happened. But anything that happens after randomization is not considered, okay? You intended to treat people with whatever you assigned them to, and that's how you analyze it. It really reflects what happens in real life when a patient goes to a physician with some problem. What does the physician do? They diagnose it, and then they give a prescription. They prescribe, you should take this drug, you should have this injection or whatever. And what happens? The person may do it, the person may not do it. The person may do the wrong thing, may take a different drug or will do whatever. And so the intention to treat principle is to say, even though we're doing a very artificial type of experiment, we really wanted to reflect a little bit about what real life would be like if this treatment is approved for that condition, okay? The issue here is that if everybody has to be analyzed, then what does that mean? Analysis means what? It means looking at the relationship between the intervention and the outcome. You have to have the outcome. So you must not have missing outcome data. This is the huge, crucial issue of how do we handle when we have missing outcome data. And we'll get to that point later. Now, another thing that is typically done is to do what's called the subpopulation of the per protocol people. And per protocol is a subset of this ITT, so it's the people randomized, but it's those that completed the study without any, some people say any protocol violations, but many people say any major protocol violations. If it's a little minor one, it's okay. But if it's major, you don't include them. So it's a subset of people, right? And why do you do that? Well, because you expect that the people that complied with the protocol are those that got the benefit of the intervention. And so therefore you'll have a stronger effect than if you did it in the intention to treat. Now, these are two well-defined groups. There are other subpopulations that are not so well-defined, and it's some that different investigators define differently. One of those is the adherers. It's again a subset of the ITT population, but it's those that investigator decides, well, they received sufficient amount of the intended intervention. If they were supposed to take, some medication twice a day for 12 weeks, for example, well, if they took it once a day, but they took it for 10 weeks, they got enough of it. Well, the clinician decides what is sufficient, and that's why it's arbitrary. But they just say, well, they got some. You'll get some effect in those, but it's just another group that you may want to look at. There's a second one that's also hard to define commonly across studies, but it's those that are called responders. And responders is, again, another subset of the ITT population, but that the investigator decides now they had an adequate response to the interventions. So this is used quite a bit in like in vaccine studies, because they expect that some people that are, let's say, immunocompromised may not get an adequate antibody development, even if they got the vaccine. So they have not, their body does not respond well to the intervention. So if they don't get adequate antibodies, then let's say you're studying whether they get infected or not, they're more susceptible to infection. So therefore, they're not really good people to evidence the efficacy of the vaccine, because they themselves are not a good body for proving this. Again, this is very subjective. It's up to the investigator to decide what is an adequate response. And so this varies, right? And it could be for other types, not just for vaccine studies, but for other studies as well. So these are 4 groups so far that are subsets of or the ITT population. There are 2 other groups that people often study that are different. They're not subsets of the ITT population. They're subsets of the study subjects. And that is the as treated and the safety subpopulation. So as treated is a subset of the study subjects analyzed according to the intervention received. So this is, you may have had people that were randomized to A and B, but some of the A people decided, oh, I don't want to do A, I'll just go and do B. And they themselves cross over to the other one and do it. Or the staff decides that we're going to do this, or by mistake does it. So some people decide that they don't want to take something. So they end up, if it's a placebo-controlled study, they may end up being in the placebo arm if they were assigned to the intervention and they don't take it. So you just analyze people according to the intervention they received. The safety group, they call it a safety population, is then a subset of the as treated one, because they now are determined to be, well, they got sufficient amount of the dose so that we will study the adverse effects in that group. So people that got, when you study the safety, you want to have the people that took whatever thing. So that is a subset of the as treated one. And you want to look at the toxicity, laboratory evaluations, et cetera, in that safety subpopulation, okay? So what is the implications of this? We have different analysis populations. The analysis of the study involves, as I said, the intention to treat. That's what you're supposed to do, okay? But people do all of these others. What is the impact of doing all of these others? Well, the effect sizes vary by the subset analyzed. The intention to treat, if you're doing a superiority study, is going to be the most conservative one. Your effect sizes are going to be smaller. And this is because you're providing estimates of effectiveness. A per protocol is expected to have larger effect sizes, and you're really looking at efficacy, right? It's people that adhere to the protocol and you get in the ideal situation. So we call that the efficacy evaluation. So people tend to do both of these. And then some people will do these adherers and responders. Remember, they're a little bit more subjectively defined. And the effect sizes then are going to be the smallest for the ITT, largest for the per protocol, and somewhere in between for these adherers and responders. Okay. The as treated and the safety groups really ignore randomization. So, if you ignore randomization, as I said yesterday, you're really analyzing a biased, non-representative, convenient sample, right? So if you're doing that, you cannot, when you report as treated and safety analyses, usually it is not valid to report statistical inference. You don't put confidence intervals, you don't give p-values. It's considered an exploratory sensitivity and is treated and discussed like that way in the paper. Some authors don't, but they should do it that way. So the implications of the intent to treat principle on the analysis is also important. So if you're including all randomized subjects, as I said, you need to have the primary outcome, so the missing outcome is problematic. But the other implication is that the analysis is what we call a crude analysis or unadjusted simple analysis that only depends on what type of hypothesis you had, what was your randomization approach, and what type of outcome variable do you have. So statistically, a clinical trial has only one permitted two-sided test, okay, and usually at the alpha of 0.05. So what about adjusting for other things? You say you're used to doing all these adjustments and models, et cetera. What if you have baseline imbalances? The randomization did not work perfectly and you just, for bad luck because you had a small study, you have baseline imbalances. Or what if some of the outcome is affected by time variant covariates, like the blood pressure is changing over time or their glucose level is changing over time? What about potential confounders, right? What about people taking other procedures or doing things outside of your study and getting seen by another physician or deciding to do whatever they wanted to do? And what if they don't comply? Your staff did not comply with the protocol, or the subjects did not adhere to the medication. They missed the visits. Somebody got unblinded and found out what they were doing. What do we do with these sorts of things? Do we include them? Many people do that, and those are considered secondary analyses, okay? Secondary, exploratory, or trying to get an in-depth, better understanding of the effect of the intervention. But as I said, secondary. You're going to do it. The primary is always that crude one. Should you present those per protocol adherers as treated? It's up to you. It's up to what argument you want to make. You may say, oh, well, the effect size is already, usually people don't do it if they get a significant, strong result for the ITT, they ignore the other ones. People, if they get a weak result for the ITT, because the effect sizes are bigger in the per protocol and adherers, or they will report those. The as treated, I said they should not be reporting, right? Because it says ignores the randomization. But some people still report it, right? Why do they report it? Because they have an argument that they're trying to make, right? Which is to convince the reader that this thing is effective. And so be aware that people will try to do these things. What about the secondary outcomes? Yeah, we usually don't have only just one outcome. We have multiple secondary, tertiary outcomes. Maybe we have things at different time points. And so we have recurrent events, for example. What do we do with we have recurrent events? Should we analyze those instead of just one event or one time point? What about secondary analysis and subgroups? Do we have to do all of these things by ITT? ITT is usually the recommended way to do all secondary analysis as well. However, many people don't. Why? Because they want bigger effect sizes, and they want to be able to claim significances and so forth. Be aware that, as I said, you have one test that is the one alpha that you have. After that, everything else is really exploratory. Many people like to argue, oh no, wait a second, I pre-specified that I was going to do this secondary analysis in my SAP. Since I pre-specified it, then I'm allowed to do the alpha, another test, etc. And it's sort of a valid argument to do because you thought about it before seeing the data. But if you do multiple secondary tests, you should consider adjusting for multiple testing, right? What about formally ignoring, analyzing, and presenting safety data, process data, quality of data? This is things that we should actually do talk about. Many people do mention safety, and they mention the safety analysis, even though they recognize that the safety analysis is violating the ITT, the randomization. So they will report, but they still report it because it's important to report. But many people do not talk about process data. They don't talk about what we call the dirty laundry of a study. Like, oh yeah, we had so many errors and we had so many queries and the data, it was not totally well done at clinic A, this, etc. So, we had inconsistencies, all of this stuff of data problems, etc., that were fixed at the end, nobody mentions, right? Because they think it's not important. It's usually mentioned if the regulatory agency asks for it, that's when it's mentioned. But in a manuscript, it usually is not mentioned. The next thing I want to talk about is when do we do analysis? And you recall yesterday we talked about the stages of a clinical trial, from proposal and funding to the definitive analysis that we just talked about. And we have analyses that are being done. Well, we have analyses that are done at multiple time points, but they differ in the type of analysis that they are. So the one that's done as the definitive analysis stage is called the end of study. This is the main analysis. This is what ends up getting published as the result of the study. The others that are done during the conduct of the study are called interim analyses. And we will talk about what are these interim analyses that we do. So you may or may not be familiar with the consort statement. The consort statement is a group of editors of journals, clinical journals, that have developed guidelines for how clinical trials should be reported. And that is what things should be mentioned, etc. And this keeps getting updated, but in 2010, they had things that must be reported included. For example, they have something like 18 considerations or whatever. So I'm just highlighting the few that are related to statistics. And they require people to mention in the paper the methods used to compare the groups for the primary and the key secondary outcomes. So secondary outcomes that are being reported, you must mention the methods and the methods for additional analysis. So like if you did subgroup analysis or you did any modeling, etc. And when applicable, explanation of any interim analyses and stopping guidelines. So you talk about the interim analysis as well as the end of study analysis, okay? and this is important to report. Blanket statements that people put in some types of journals, like in the section of statistical analysis, people will say, oh, for continuous data, we use means and standard deviations, and for frequencies, and so for categorical data, we report frequencies, and the tests that we do are for continuous are t-tests, that's in chi-squares for the categorical one. Those sorts of generic, meaningless statements are not appropriate for here. They have to say what you specifically did for the primary outcome. What did you do specifically for this other secondary outcome? What did you do specifically for the subgroup analysis? So generic statements are usually, in general, I don't like them, but people still continue to do those sorts of things. But here, the concert guideline recommends that you be specific about what was done for the different outcomes and analysis. So, as I said, the primary analysis on the primary outcome is dependent on the type of variable and the type of hypothesis as well. So how does it depend on the type of variable? What sorts of variables do we have? We have multiple types of variables. We have the baseline variables that are prior to randomization. Maybe at the screening stage, we consider those as fixed covariates that may be considered if we're doing modeling. They're used primarily to check eligibility and also to check on whether the randomization balanced the groups at baseline. And it also enables us to do subgroup analysis, because we may subgroup on gender, we may subgroup on age group, or we may subgroup on level of severity of their condition, etc. Then we have follow-up variables, and these are the ones that are collected after randomization. And so they may be time variant covariates, like your blood pressure changing over time, or your glucose level, or whatever. They're useful for checking compliance, adherence, for monitoring the participant care. These are things that are part of the study, looking at adverse events, etc. But it also includes the primary outcome variable, which is, as I said, what we call the analysis of the trial. We also have secondary analyses of that primary outcome. So we may have models that we do, and whether we want to do those or not depends on whether we want to adjust for baseline imbalances, or sometimes people even adjust for post-randomization characteristics. And we'll talk a little bit more about that. But we also have other outcome variables, right? Secondary and tertiary variables. So I was going to show you some examples of statistical analysis section. And one of those is from the Pfizer vaccine for COVID paper, which came out in December of 2020. And here they talk about first the statistical analysis. Notice here they don't have this stupid generic thing of we use the t-test for continuous, and we use this for that, and we use that for that. This is very specific, and it says safety analysis. Who did it include, and how did you do it? And then the analysis of the first primary efficacy endpoint included participants. So who did it include, and how did you do it? So vaccine efficacy was estimated this way with the incidence rate ratio, and they give you detailed ways of how they dealt with it. They also mentioned that we had to compensate for an interim analysis, and so therefore that modified the alpha a little bit, and this is how we did it, okay? So this is usually what is done in a serious reporting of a randomized controlled trial. Other people still tend to do reporting that really is not considered the best way of reporting it, but it's okay. This is a paper that appeared in the BMJ on looking at thromboembolism in older acute medical patients. And they call it statistical analysis, but they start off with sample size. Sample size is not analysis. Sample size is a design issue. That should be under methods, not under statistical analysis. You may want to call it statistical considerations of design or something, or put it in design early on. I verified it did work, that they had 400 patients per arm, but the primary efficacy analysis was carried out on all randomized patients with an available efficacy assessment. And they also mentioned how they did the safety analysis. So again, the details of the safety analysis and the details of the primary efficacy analysis. And they also mentioned they only talk about in this case, they only did the primary, they had no secondaries. But if you have secondaries, you mentioned secondaries. The other thing that determines besides the type of variable, besides the outcome variable, is the type of hypothesis. So we've talked yesterday about the superiority hypothesis, the non-inferiority hypothesis, and the equivalence hypothesis. And we talked about how we go about specifying these and the parameters that are used to specify what is it that you want to detect. And here it's the smallest clinically meaningful difference. Here, capital delta is the maximum tolerable difference for non-inferiority, and gamma is the maximum tolerable difference for equivalence. How does this affect the analysis? Well, it affects the analysis because usually the little delta is greater than the capital delta, which is greater than the gamma. And so we will see that as the difference between the null and the alternative is smaller, the sample size gets bigger. And so the sample size for an equivalent study is bigger than the sample size for a non-inferiority study, which is much bigger than the sample size for a superiority study. Okay, and so sample size will affect the analysis. Why? Because if the sample size is large, then there's balances, right? Randomization works. If the sample sizes are small, then you will have likely imbalances, and you may have a smaller effect size. The power of the test is also going to be different because it depends, of course, on the sample size. So that's how the type of hypothesis will affect the analysis. The final thing that affects the analysis is the method of randomization. Okay, and one of the last ones. And simple random allocation doesn't affect it. Of course, that's what we're basing it on. But if you do some restrictions on the random allocation, you may be affecting the variance of the primary outcome, and you may have to take that into account in the analysis stage. Usually people argue that blocking or permuted block, like we discussed yesterday, does not really affect it. Some people, there's some papers that argue that it does affect it somehow. But many people do not take that into account, or sort of basically ignore it in the analysis. When you do stratified, you actually increase the precision, and so people like to take that into account. And then in matching and optimized randomization, you must take that into account. Specifically, when we do clustered or group randomization, we have to take into account what's called the design effect, and that does affect the how we do the calculations. That's all. But the final thing, and the most important one that determines the type of analysis, is the type of outcome variable, right? The distributional property of the outcome variable. So if it's a continuous outcome variable, like means or changes in means that people do, we always have the analysis is a very simple test. Why? Because it's crude. Remember, it's unadjusted. So the primary analysis is a simple test. You may be disappointed to hear that, but that's exactly what it is. It's a very simple test. So if it's continuous, it's going to be, the unpaired t-test or one of the standard non-parametric two-sample tests, right? Mann-Whitney or Kolmogorov-Smirnov. If it's categorical or an ordinal scale, we're looking at frequency distributions at a given time point. So you may do Fisher's exact, or if you have enough sample size, a chi-square test. If it's binary, we're looking at the proportions and we look at relative risk or risk differences. Again, Fisher's test or chi-square test. And many times people like to do time to event or survival outcomes. This is very common in chronic diseases to look at survival outcomes for two reasons, because you're going to have sensory observations is one, but also if you're looking at the occurrence of an event, which is a binary outcome, if you miss people because people drop out of the study, then you have a missing outcome. And missing outcomes, we say were problematic. So people do time to event, because if you time to event, when people disappear, they withdraw, they get censored at that time point. And so you don't have missing outcomes. They just were censored, and you include them in the analysis. So you can say, we're still doing intention to treat, and we don't have to go about imputing missing values, for example. So, but if we have time to event as our outcome, then we do this standard log rank test. So these are very simple tests, right? Now, when we're going to do analysis, what are the typical stages of analysis? So the stages are, first, every paper starts off with the famous table 1, which is a subjective comparison of the groups at baseline to assess randomization. Did it work? Yes or no? There should not be any p-values on this, but people in the past tend to do it. Nowadays, most journals will demand that you remove the p-values from table 1. Then comes a subjective assessment of process variables to assure the study was done well. And this, as I said, many people do not present it at all. They just they may put one sentence that the study was conducted under GCP and everything went well, or they may not say anything, right? But this was a good study. We had very little attrition, people adhered, and then they mentioned those sorts of things. Then comes the descriptive analysis of the primary outcome. So they may say, the proportions are this, or the means are this. They may do a Kaplan-Meier curve, for example, if it's time to event data. Then comes the inferential analysis of the primary outcome. And this is the main results. It becomes usually table 2. And it's calculated crude estimate of the intervention effect. Then they may assess if it's clinically meaningful. So clinical meaningfulness is subjectively assessed, right? And that's why people don't like to say this was meaningful, because that's subjective. That's why they only talk about significant. But significance is a statistical thing. It's not a clinically significant. So if it's clinically, it is meaningful or important or whatever, it's not significant. The word significant is a technical term that should be reserved for a test of hypothesis, a formal test, statistical test of hypothesis. If it's a clinically meaningful result, then they say, oh, it's not due to any bias. And so if it's now not, if that estimate is meaningful and it's not due to some bias, then now you're sort of entitled to go ahead and test whether it's due to chance or not. And you calculate the p-value, determine if statistically significant. And that doesn't stop there. Once you have statistical significance, you have to put it in context. What context? Clinical context. So where does the statistician come in? The statistician comes in at this stage here, but also at this stage on ruling out biases, on calculating this, and on doing all of this descriptive analysis as well on this, and preparing the tables, of course. But in the interpretation, of subjective things. That's where the clinician comes in, putting it in context and subjectively assessing and subjectively assessing and subjectively comparing. That's what the clinician or investigator does. The statistician comes in into the formal testing, the ruling out of bias, and in the descriptive analysis and construction of things. Okay. So let's look at an example. This is one where we saw yesterday, again from the Pfizer vaccine trial. This is their table 1. We see no meaningful differences, and we notice that there's no p-values. Now, another thing that comes in between, that's not really analysis, but between table 1 and table 2 of results is the consort diagram, we call it. And the consort diagram is a flow of participants in the study. And again, this is from the Pfizer one, that basically starts with screening of participants all the way down to who was analyzed and what happened to them, what happened to everybody. And specifically after randomization, we want to know who was excluded from analysis, because remember that the ITT principle says once you're randomized, and they had 44,820 screened, but they had only 43,548 randomized. After randomized, that should be the people that are ITT, everybody randomized. But there are some little caveats here that this group dealt with, and they said, well, we had this many randomized, but only this, so about 100 less received the 100 did not receive the injection, so they really should not be part of it. They just did not get it for one reason or another. And so they removed those. But they also limited it, and in table 1, the people that they're comparing is the ITT group, and it's those that had reached their 2-month follow-up. And so this is something that their study decided they wanted to do. So this is what they considered as their ITT. But when they're reporting the analysis, they're wanting to report the analysis on those that received 2 doses. So some people only received 1 dose, and so they're reporting it on a fewer people. When we go to their efficacy results, which is table 2, they actually give it for even fewer people, okay? Because they wanted to eliminate those that were not eligible because they already had baseline infection, right? And so therefore they wanted to say these people were not eligible, so we removed them from it at that stage. So it's smaller than the ITT, and they report this. But then they also reported for people that had prior evidence of infection, and this is a group that is actually bigger than the ITT. So, which is the ITT analysis here? It's this top one here, and basically they showed the main result here, of course, they got is that they had in the vaccine group, they had 8 people that had infection, and in the placebo group, they had 162, and they had therefore what's reported in the media, the 95% vaccine efficacy. In fact, this calculation is as simple as 8 divided by 18,198 over 162 divided by 18,325. So it's just a simple ratio of proportions, rate ratio, okay? And then 1 minus that, of course, because that's going to give the efficacy, not the incidence of it. But then they do all sorts of little bit more complicated. They do Bayesian analysis to get certain other, the p-values, et cetera, and so forth. So the crude analysis then comparing the 2 RCTs is under the intention to treat. And that's simple, as we've seen. And that table 2 is as basic as can be. There's nothing complicated about it. Now, there are complications, and we need to deal with these complications. One of those major potential problems, and I'll say that again because it's important, is that the outcome variable is missing. So it is of the utmost importance that when you are conducting the study, when you're designing the study, you make it in such a way that it is not easy to miss the outcome. So you make it easy to obtain the outcome. And people then like the outcomes to not be measurements because people get lost, etc. They like it to be a survival outcome, as I said. But many times, sometimes the outcome is missing. And if it's evaluating the health status of a person, you don't want that person to disappear and not be measured. So if you're doing a study that is measuring something, as opposed to time to event, you could have the data missing. And so in missing data analysis parlance, we have different types of missing data. We talk about the missingness mechanism, and it could be an informative reason, which is a biased one. So we call it missing not at random. And we may have at least what's called missing at random. Missing at random is that the reason it's missing does not depend on the value. So it is manageable. Ideally, we want it to be missing completely at random. And what's the difference between missing completely at random and missing at random? I'll give you an example. In a study of obesity, if you're going to weigh people and the outcome is the weight of a person, people that are, let's say, more, and it's a weight change one. So people that have not dropped or maybe have increased their weight are more likely to not show up to be weighed. So now the reason it's missing is dependent on the weight or the weight change. So that is very informative. Now, if it is something that does not depend on the value of it, so suppose that it is, people do show up regardless of their weight change or not, but some people show up and others don't. And maybe we were doing the study in a university college setting, and it was the staff of the university that was participating in the study. And some of the employees were not allowed to leave at lunchtime to get themselves weighed. And so the people that this was not dependent on their value, it was dependent on their job. And the employees that were staff that were not allowed were maybe lower socioeconomic status than those that were allowed to come. And so now the missingness was not dependent on their weight change, but it was dependent on their job. And so that would be missing at random. But to be missing completely at random, it should have been, the reason this person did not come was just because they had a car crash that day, they didn't come to work that day for some other reason. It doesn't really depend on anything else. It was just, this random situation. Okay. That's a lot harder to prove, right? So usually we try to prove missing at random, and that allows us to handle the missing data. What's the best way to handle missing data? Well, one of my professors said, avoid having it. And that is actually, may sound like a joke, but that's the best way, is try to design your study, conduct your study in such a way that you minimize and almost avoid totally having the missing outcome data. Otherwise, people say, they throw out some figures, in a randomized trial, you should not have more than 0.5% missing data or 1% missing data. These are totally arbitrary numbers and percentage. Or you impute the data. And impute, sounds very fancy technically, but basically what you're doing is you're making up the missing data. And anytime you make it up, it's problematic, right? So we have to be aware of the problems with that. There are many methods to impute in a prospective study, as in randomized trials. We may be able to, if we're measuring the outcome along multiple time points. We can have the last value carried forward. We could use regression approaches, estimated based on their previous values extrapolated, or taking means of the values of other people that are similar. There's all sorts of different complicated regression-based approaches that people come up with. Those reduce the overall variability. So in order to not do that, nowadays we do that with multiple imputation. So there are ways to handle it, but be aware that if you have a large proportion of missing outcome variable, I don't care how fancy your multiple imputation is. I bet that the journal reviewers will say, this study was so poorly conducted, they had X% of missing outcome data. They imputed it, but if they did not get outcome data, they had so many missing. This study was not well conducted. I think we should reject this paper. It's a meaningless paper. So be careful. Missingness here, yes, we can do all sorts of fancy stats to deal with it. And we argue that it's at random, and we can show that it's at random, but still it is problematic in how the others will view. And when you do a trial, you're trying to change clinical practice. And if people don't believe your study because they didn't think you did it well, no matter how well you imputed and handled it, you are in trouble. Okay. So avoid having any is the way. Okay. All right. So finally, we worry about, do we have adequate power for the analysis? And this is where the sample size is something that has already been determined at the beginning of the study, at the design stage. Maybe you did an adaptive design where you looked at the data halfway and decided whether you needed to increase or change the sample size. That is many times problematic because funding agencies don't like you asking in the middle of a funded study, give me more money because I need a larger sample size, or give me more time to recruit, and so forth. So you do a reasonable estimate at a design stage of the sample size needed for what? For many times we either talk about power or we talk about precision. However, in a clinical trial, it's almost always power, because what we're doing here is comparing two interventions. So we're testing. And so we are always in this hypothesis-driven testing situation. So it's power that determines it. It depends on the research design. As I've said, the level of measurement of the variable, type 1 probability, type 2 probability, and the distribution of the statistic used given the study randomization design. So the randomization design does impact the sample size calculation. If it's simple randomization, this is what we normally assume is guiding things. We may do stratified randomization, which often gives us lower standard errors. But if we do weighting, then we do increasing standard errors. And if we do cluster or group one, this is a two-stage process of selecting participants. You do have a loss of effectiveness, and we quantify it by the design effect. What is a design effect? This is just a terminology that sampling statisticians call variance inflation factor, but it's exactly the same thing. It's basically taking the amount of correlation there is within a cluster or group, the average group size, M, and you notice that this is then going to be something that is greater than 1. And to the extent that it's greater than 1, it increases or inflates the variance that way and affects the sample size calculation. How does it affect the sample size calculation? Well, the conceptual formula for every sample size calculation for testing is given down here. And that is that your sample size is going to be directly proportional to the power. The more power you want, the higher the sample size on the variability of the outcome. So the higher the variability, the higher the sample size. So therefore, if you inflated the variance because of the design, then you're going to require a larger sample size. Your significance, if you wanted a smaller significance level, like 0.01 as opposed to 0.05, you would have a larger sample size because it's inversely proportional to that. But usually everybody sticks to 0.05. Nobody ever changes this. Power may be 80 or 90%, but nobody goes beyond that or below 80. The thing here also is the difference to detect. And remember that difference to detect was that little delta or that capital delta or that gamma, according to the hypothesis. And that is important. Those are small numbers, and it's inversely proportional to the square of that. So the thing that has the major impact on the sample size is that difference you want to detect, okay? And these differences then are those subjective things that are clinically meaningful or clinically important to detect, okay? And that's what drives it. And it's the delta here and non-inferiority, or it's the, I guess I should have put here, this is a mistake here, should be not equivalence, but non-inferiority. We have then delta as the largest difference clinically tolerable. Okay, so that's what other considerations do we have to worry about in the analysis, right? So we said the analysis is basically simple and crude. It depends on the hypothesis. It depends on the randomization scheme. It depends on the type of variable. Does it depend on anything else? Do we do other things? Well, we said the sample size is going to be affected by, do we want to adjust for missing data or loss to follow-up or drop-ins and dropouts, for example, people that cross over. So that's going to affect the analysis, or multiple testing adjustments, corrections for multiplicity of independent tests, or if we do repeated significance testing of accumulated data, and we'll talk about that in a second. Other considerations, if you're doing a finite sample, in a finite population, you may have to adjust your sample size for finite population correction factor. This is not often done because most people argue that the sample sizes of our populations are infinite. Other considerations we've already mentioned, the design effect. Don't be fooled because very small correlations can have a big impact on the side effect depending on the sample size per group, for example. So complex designs may also be ones that you have to adjust for. And then we tend to do complex analysis in this, okay? We may do adjustments for covariate imbalances or do other types of modeling, but it is usually a secondary type of analysis, okay? So the crude analysis, as I said, is already mentioned here under intention to treat. It's very basic. I've already mentioned before that it's either for binary, it's a Fisher's exact or chi-squared, or if it's a time to event, it's the log rank. But we have some complications or interpreting the primary analysis outcome. What happens if we get a negative result? If we get a negative result, we should still publish it, okay? We should rule out poor conduct and the poor quality of the data, biases. People like to calculate post-hoc power. I'm not a big fan of that. I think power is a probability, and probabilities cannot be calculated after the fact. You calculate probabilities before the fact. So then you have to be very careful. If we had done this, and if this had been what we wanted to detect, we could have detected this with this power. It makes really no sense after you've done the study to do it. So I don't particularly like to do this. Now, there's other complications to the standard analysis. So what if we have more than two arms? We've been talking only about two arm designs. Sometimes we may have a control arm, and two different levels of dose. So now, what is our hypothesis? What is our alpha? Do I split the alpha? So I have one alpha for dose A versus control and dose B versus control. But what if I want to compare dose B to A? So then you have to sort of decide how you're going to share the alpha, or what you're going to do, or you're going to do a hierarchical testing. You're going to do a global test. If there is a significance in any difference, now I will test that. And you may or may not want to do it. So people are very specific in how they share the alpha. And if I have three different drugs, do I want to compare A to B and A to C and B to C? So which ones? How do you do it? So those are little complications. There's not any guidelines really. Is this a standard way of dealing with sharing alpha? Remember, you have one alpha to share, okay? What if we have more than one primary outcome? Some people have two co-primary outcomes. And I mentioned yesterday that the Food and Drug Administration in the United States has standard ways of saying how you should share the alpha and how you should interpret the study results. If you call them co-primary, you must have both of the primary significant to say that the intervention works. So therefore, people say you can have multiple primaries is different than co-primaries. And you can, again, share it by saying, oh, we're going to split it half and half, or we're going to say this is more important. So this primary is really primary. So I'm going to give it 0.04. And this one, it's less primary, but it's still primary. I'm going to give it 0.01. These are arbitrary things that people tend to do, or they do it sequentially. Then I'll test this one, then it becomes really a secondary. If we have to impute outcome for too many participants, like 4% or 2% or whatever, what do you do? How do you report that? You should be transparent and report it. If you want to adjust for baseline imbalances, you do the modeling, etc., and then you report those models. Some people like to analyze change from baseline in a single arm. Well, that violates randomization. That's invalid. It's actually something that people do that they should not do. Okay. Now, recall that I talked about the typical stages of analysis, and I did not mention one of those, which is that we do the secondary analysis, right? So I've talked about the typical stages of analysis for the primary, but then we also have secondary outcomes, subgroups, and covariate adjusted primary outcomes. What are these analyses for? These analyses are usually for two reasons. One is generating hypotheses, and the other one is looking at consistency of the result of the primary outcome. So it is consistency that you're looking at. It is not necessarily testing new things, right? So the secondary and subgroup analysis is specifically looking at the subgroups are in specific subsets of participants. And that's when they try to look at people with different risks or different demographic considerations, baseline variables. This is when you look at the per protocol, the untreated, the adherers, the responders. And it can provide useful information. I don't say it's not valuable, it's very valuable. You can get the consistency of results, or identify subgroups. But there are possible harms in doing something like this. So the effects in subgroups should not be assessed with separate tests for each subgroup. So you don't say, oh, does this work in the age group such and such, or does this work in the ethnic group such and such, or does this work in these and people with more severe or not? You don't do separate tests in different categories of subgroups. Multiple tests increase false positive rate. And if you want to do a test comparing the effectiveness of intervention in a subgroup, you do a test of interaction. That's the only valid test that you should be doing. Okay. Now, you should only do these if you have pre-specified it. So it's not post hoc ones. you have to do it pre-specified. So you have to put that in your protocol. It's ideal that it's only be few people and that you have some reason for doing it. You don't just do it on people's zodiac symbol or whatever, religion or something. You do it on biological plausible situations. And if you're going to be doing this, you should actually have enough power to detect subgroup differences, and this interaction test often don't have enough power. So interpret with caution and only use it to generate hypotheses that will need independent testing. So guidelines for this, you have to interpret them in light of the totality of pre-specified subgroup analysis, because you probably have done multiple of these, okay? And if you've done them post-hoc, you should label them and address the multiple comparisons issue, okay? Nowadays, people are doing shrinkage analysis, which is a Bayesian technique to sort of draw information from the overall effect to look at the subgroup effects. And that's something that is important to consider as well. But whenever you see subgroup analysis, beware as the user. I sometimes think that people do the subgroup analysis to torture the data so it confesses to something that they're interested in finding an effect in a particular subgroup. So the last thing I want to talk about is a consideration in clinical trials. And that is that, as I said at the beginning, is that we often do not have the luxury to wait to do the analysis at the end. Yes, we do an analysis at the end, but we are forced by ethical reasons to look at what's called interim analysis. And it could be that the reason we're driven for this is because what we call lack of equipoise. We start the study when there's equipoise, but maybe during the study, the equipoise is gone. Once intervention is seen to be better conclusively, then why should we continue? Or there's some safety issues, or the study is a worthless study. It's futile to do. So did this happen? Is this a common occurrence? Well, yes, it is relatively common. Here's a result of a study on HIV infection and early antiretroviral therapy given to the partner of somebody who was already infected. Does it prevent? And the Data and Safety Monitoring Board recommended that the results of the study be released on the basis of data collected as of a given date. So they stopped it. And they specify here, according to the monitoring guidelines, they were based on This exceeded this, and therefore they ended up stopping the study because they already had shown it was efficacious. So it was done for efficacy because equipoise was no longer there. Here's another example where the trial was stopped early for safety reasons. This was a much smaller study, and they were finding out that the rate of severe hypoglycemia was higher in the intensive therapy group than in the conventional therapy group. So we cannot proceed. This is not safe. You got to stop the study. And the board recommends stopping the study. Here's another one. The trial was futile. They just, patients were not, this was a surgical study, and there were no significant differences early on. Even if they had continued, they would be inconclusive at the end, and the study was stopped. Now, all of these studies being stopped are stopped by a recommendation from the Data Safety Monitoring Board. It's never an investigator who's looking at the data because they don't want to stop, right? Because if they stop, they have to return money, that funding money. So it's always that external group, and the Data Safety Monitoring Board only recommends it. They recommend to the sponsor, and the sponsor is the one that decides whether they want to stop the study or not. So as I mentioned yesterday, when talking about DSMBs, the rationale for interim analysis is driven entirely by ethical reasons. And there's three types of considerations that come into this: the analytic ones based on information, administrative ones based on the study conduct and progress, and subjective ones which are external to the study, other studies coming up with things. And so you have issues of unacceptable safety problems, or you have effectiveness has been shown, or it is unlikely possibility of results, or it's futile to go on because of maybe low event rates or smaller difference than expected, et cetera. Administrative ones was poorly conducted study, or other results from other studies. Now, these are subjective things, administrative and subjective ones. I'm going to talk about how do we handle these here, because there's a base on information. So primarily based on the testing of the primary hypothesis. So remember that the type of primary hypothesis testing is basically crude under ITT. It's very, very simple, right? And those simple tests are either Z tests asymptotically, or they're chi-squared 1 degree of freedom tests asymptotically. And we know that the chi-squared test square root is this, the Z. I'm going to talk in terms of Z. So what happens when we're doing interim analysis and monitoring the study from a hypothesis testing situation? Well, actually, what we're doing is we're testing the hypothesis, the primary hypothesis, every time we've specified we're going to do it. So let's suppose we have a study that's going to go on for like 3 years, and every 6 months, the Data Safety Monitoring Board says, I'm going to look at it. And they start at year 1, and so 12 months, 18 months, 24 months, 30 months, and then finally at 36 months, they will do the final analysis. So they have a final analysis, and they have 4 interim analysis. So suppose this hypothetical example was taken place. So I have these 4 interim analysis at times T1, T2, T3, T4, and I have the final one at T5. So we're doing it at the 0.05 level. So what if I did the test at 0.05 and I had 1.96 as my critical boundary, and the study starts, and after 1 year, this is the Z value I get, fine. Then I continue the study, and because I haven't crossed the boundary, we continue. And the next time, oh my God, it gets to no 0 effect. So should we stop the study? Well, no, we are looking at the potential for superiority. Maybe we haven't established a criterion for futility yet. We're to establish a criterion for superiority. So we continue. We're now, we're getting closer to it now. So, things are getting better. Do we tell the investigators? No, you don't tell the investigators. They should not know this because they may change what they're doing and make it bias the results. So you don't tell them anything. But now at T4, so now this is at 2.5 years, you get to crossing the boundary. Should we stop the study? We reached statistical significance. Well, no, you have to pay a penalty for having done the test multiple times before. And it's not independent tests, they're based on cumulative data. And so the actual, if you do 5 tests at each at the 0.05 level, your overall level of significance increases to 14%. So you don't want that to happen. So you need to adjust for these multiple looks of accumulated data and correlated tests. They're not independent tests, so Bonferroni adjustment is out of the question here. So how do we do about it? Well, this is something that was determined a while ago with sequential study analysis. So this was because this is a sequential study. You're analyzing the data repeatedly. And when you get to a stage, you either terminate or you continue. This sequential testing originated in quality control in 1929, but it wasn't until the 1940s where Abraham Wald came up with the sequential probability ratio test. Peter Armitage came up with repeated significance testing and how to adjust that alpha for it. His test required that his procedures that the number of looks be pre-specified. We're going to do 4 previous looks, so we're going to do 5 looks total. etc. This gave rise to group sequential designs of equal-sized groups of patients or of information are analyzed at pre-specified number of times. So these designs said that after results are obtained at a given stage, at the kth stage, you calculate this accumulated standardized test statistic, ZK, you compare it to critical value, and if you're below the critical value, you continue. If you're above that, you stop and you reject, okay? And this is basically coming out from Brownian motion stochastic processes analysis, because he was able to show that you have this accumulated ZK statistic can be expressed as the sum of independent normal increments. And so this was the breakthrough that was needed to be able to make adjustments to the boundaries for multiple testing of accumulated data. So some people came up with how to adjust, and Stuart Pocock in the UK said, well, one way to do it is to say how many tests you're going to do and have a constant value that takes into account K. So his critical value was based on the number of looks, and for 5 looks, it's 2.413. So Pocock's boundary then becomes 2.413. And this Z4 doesn't cross it. So according to Pocock, I would continue. And so if we use Pocock's and continue, and we get to the end, we get 2.02. Now the study ends. I, the statistician, now go to the investigator, unblind them and tell them the study results. They're waiting there, biting their nails, expected to hear what the results are. And I say, well, I'm sorry to say that the study was not significant. We got a Z value of 2.05. And he said, wait a second. I took a course on statistics. I know that 1.96 is the critical value. Why are you telling me that 2.05 is not significant? Oh, it's because we have to adjust for the interim analysis that we did, blah, blah, blah. And we're using the Pocock boundary. Says, we don't have to do that. We don't have to report that we did that. We just report the final one, it's significant. So people did not use Pocock because of that, because it's harder to show significance at the end if you don't stop early, right? So then came other boundaries that were suggested by Hay-Biddle and Pito that said, make it very difficult to stop during the study. You have to have a Z value of 3 or more. But the final value is one that is adjusted to the number of looks, and it's closer to 1.96. So for them, when they did their calculations, that the 5th value is 1.99. You just have to be very, so the 2.05 would be significant there in that case. So this is what Hay-Biddle has done. Others have argued that you can even do it higher, and you don't even have to adjust it at the end. You can use 1.96 at the end. The problem with the Hay-Biddle-Pito is people said, well, you really are making it so difficult to stop even closer to the end when you have a lot of data. You're not likely to stop. So why do you even have this thing, right? So in 1979, we had O'Brien and Fleming who came up with, we should have these boundaries dependent on which look we're at. So it should be a decreasing boundary, sort of smooth boundary. And so the final one for them is 2.04, so that 2.05 would have been significant, just barely. But they have it so that it's almost impossible to stop really early on. And you don't want to do it because there's a lot of variability at that point. But as you start getting closer to the end and you have more information, you start getting to a place where you would likely stop, right? And so this is now usually one that is accepted. But remember that all of these require that you pre-specify the number of looks, okay? So in summary, these are the ones that people have proposed. There have been some adaptations. Some of the students of O'Brien and Fleming have developed new ways of generalizing the critical values. But the problem was having to pre-specify the number of looks. So a few years later, in 1983, came another breakthrough, and this is by Dave Demetz and Gordon Land, working at the National Institutes of Health in the United States. And they said we should think about that alpha of 0.05 as if it was money. And we are spending money. So if we use a lot of the alpha in an early look, we have little alpha for the end, and it's going to be harder. So why don't I just relax the requirement of when we do the test, not have to pre-specify it. The only thing we have to do is pre-specify for the next look how much of the alpha we're going to spend at the next look before we see that data, of course. So the timing and the number need not be pre-specified. The groups don't have to be of equal size. And so they came out with spending functions that reproduce the O'Brien-Fleming boundaries and the Posthoc boundaries. And this is what we use today. We use this Land-Demetz approach of alpha spending, but we tend to use it for the O'Brien-Fleming boundaries. Okay. What is the impact that this may have on the sample size? Well, very little. The O'Brien-Fleming and the Hay-Biddle-Pito, even with 10 looks, only inflate the sample size 4% or 3%. Okay. Posthoc is pretty bad. This is another reason why Posthoc is not used, because it inflates the sample size quite a bit, you know. So that's why. Have these things been used? Yes, there have been classic examples. This is used all the time. In almost every study has some stopping criteria in your interim analysis. And some of them there are other techniques as well of looking at power. We call it stochastic curtailment, conditional power. So there's many other ways, but these are more commonly used. A famous study came out in 1982 was the beta blocker heart attack trial that was stopped early. It's one of the first studies stopped early. So way back 40 years ago. And they had the O'Brien-Fleming boundaries here, and they crossed it 1.96. They had crossed really early, about 3 years before they actually stopped the study. But they had not crossed the O'Brien-Fleming boundary, and when they crossed it, they stopped the study in October of 81. 2 years after they had passed up the 1.96, and they published a paper in June of 1982, okay, or March of 1982. Another famous study is the Women's Health Initiative done in the United States, looking at hormone replacement therapy and its effect on women's cardiovascular health or coronary heart disease, or in the adverse effect of breast cancer. And they stopped the study. They had O'Brien-Fleming boundaries, different ones for superiority and for lack of safety. And for lack of safety, they crossed the boundary and they stopped the study. And so that's another one that's important. When a study is stopped early, it becomes a dramatic issue and people make a big deal about it. So this is some of the challenges that we have in analysis of clinical trials. The basic primary analysis is very simple, but the challenges come in the fact that we do interim analysis, the fact that we do subgroup analysis, and the fact that we may have imbalances because we haven't done a large study or we have missing data. We want to adjust for that. So, questions, comments? Nice. So there's two in the chat that I'm seeing. One says, in case we're doing academic clinical trials, not regulatory, is DSMB still mandatory? Yes, they're mandatory, whether it's not only regulatories that want it, it's safety that dictates it. So whenever there is a potential of safety, then you do need a DSMB. The only times you may not need a DSMB is if it's administratively not possible to do it because the study is only lasting for 4 weeks or 2 weeks, or it's a very, very short study, then there is no sense of having a DSMB. But in general, any study that is going to last more than a few months really should have a DSMB. The United States requires the NIH, if it funds their study, they don't care if it's an educational intervention, they don't care if it's a dietary intervention, anything requires a DSMB. Now, there may not be a very formal one, but it requires some external monitoring, primarily for safety and for data integrity. So yes, you do require a DSMB. That's from, and another one, thanks for the lecture. How do we calculate the inflated value in sample size for various stopping rules? I'll have to, I mean, there's software for it, but I'll have to refer you to the original papers for how they did the calculation, but it is not that easy. That I can give you the paper, but it's not easy to do. There are some software for it. I think it's called EAST. There's a group in the UK that has developed this. There may be some R packages as well for it. Okay. Any other questions? There's somebody.
Good evening, sir.
Hello.
Can you hear me? I'm Anju from.
Yes, Anju.
Yeah, I'm from Cochin. I'm a dentist. I'm not a biostatistician. It's a doubt regarding sample size calculation. Like how you stated about having a sample size based on the power, the difference, and we do a minimum sample size calculation if it's a 2-arm study. So how do we assess the rate of attrition that can occur? And do we have to increase the sample size, anticipating that there would be 5% attrition or 10% attrition? So how do we decide that? Or do we have to just state the minimum sample size that has been calculated? When we have
so usually, so good question. Usually, so one thing that I want to make clear is that whenever you are doing a randomized controlled trial, that's usually not the first study you've done on that topic, or you have previously done other studies, either at your institution or other people have studied this, and so they know what is likely to be expected for attrition. And it may be that in another study, somebody did it and they got 2% attrition, another one got 40% attrition. You had looked at your clinics, and in your clinic, people were like 8% attrition. You try to guess what it's going to be, but you want to inflate it for attrition because the necessary sample size is after attrition is taken into account, what is the necessary number that you need. So if you don't take attrition into account and you say, oh, my sample size based on the formula is 100 people, and now you have 40% that have left, now you have only 60. So you're going to be in trouble. You will not have the adequate power. And so you do want to take that into account, and you need information from prior studies. If not, then you make up a number, 10% or whatever, but there's always literature somewhere of similar types of studies than somewhere else. Maybe not the best thing, but it's something.
Okay, sir. Thank you so much.
Then Kalai, there is a question from Kalaivani Kant.
Yes.
Sir, good evening.
Hello.
So in the non-inferiority trials, like any other superiority or equivalence trial, we do have a secondary outcomes. But for a primary outcome only, we specify a non-inferiority margin.
Right.
Is this secondary outcomes are really important in the non-inferiority trial? What message it is actually giving? Should we consider this as just a hypothesis generation?
It really, it depends on what you're studying and how people are interpreting, right? because it could be that your primary outcome is a non-inferiority one, as you say, but some of the secondary outcomes are for superiority or for safety or for whatever. So depending on what it is, you may specify them as a secondary objective of a study, and you may adequately, if you have adequately powered for those, you may be able to test them. So they don't always have to be hypothesis generating. They could be ones that, some investigators like to argue, we're doing a study, we can answer multiple questions with the same data. And we're going to do, the main thing we're going to do is a non-inferiority one, but we want to show this other objective 2 and this other objective 3, and we have these other secondary outcomes. We've powered for it, and we want to test them. So yes, you could do that, but it varies, right? It's not, there is no prescription of how to do this. It depends on what you're studying and the argument that the investigator is willing to make.
Sir, I do have one more question. Heard this responders population analysis for the first time. So trying to understand, recently there was a paper from NEJM, maybe this month only it is. They have done a coronavac inactivated SARS-CoV-2 vaccine efficacy analysis. In that, they have mentioned that what they've done is they have taken unvaccinated and partially vaccinated and fully vaccinated. In the partially vaccinated and fully vaccinated group, they have removed those people who are infected within 14 days after the receipt of their first dose and second dose. Are we talking that as the responders analysis?
No. So this is, what they're trying to do here is eliminate people that were already infected, but sort of asymptomatic. So that somebody that, what they're assuming is that if you have developed infection in the first 14 days, that you already had it and that you just had not manifested it. So that, maybe 4 days before you get randomized, you actually got infected. You were not showing any signs. They didn't re-evaluate your eligibility by checking your virus status. And maybe they screened you 2 days ago and they're randomizing you today. And in those 2 days, you got infected. So that's why they want to eliminate those people, and they're calling them noise in that. So it's not the same thing. Responder is something that it's a little bit difficult to understand, because it is it's only in specific situations where they think that you are not receptive to the intervention. So for example, I mentioned the vaccine example, but suppose I take another type of design where I have a educational intervention. So, yes, you are there in the study, you are randomized to receive a particular type of intervention, but it is, and I'm going to evaluate how well you perform given that intervention. But what if you fell asleep in class? And so you did not get it. So you did not receive it. You were not responding to it. So you did not get adequate thing. Or maybe that could be a sort of non-compliant one or adhere one. So let's say a responder would be, I'm thinking out loud here, that you did not understand the lesson well, so you were not able to carry out what you were supposed to do, right? So there's all sorts of things. That's why I said I put it in parentheses, in quotes, because it's really hard to define. And people sometimes use those terms, but not very commonly.
Sorry, sir, I forgot to mention, it was not a randomized control trial. It was the healthcare workers, they have kind of cohort study. It is retrospectively, they have studied this.
Well, regardless of, yeah. but the same concept applies, right, in defining it. It's a tricky thing to define. What is a responder?
Thank you, sir.
Any other questions?
There's another one here. Is there any method to find out if number of tests can be calculated due to the limitation of cost? I don't understand the question.
Yeah. Okay.
And then there's another one. When you were discussing the who do we analyze in case of analysis based on responder, sir, give the example of immunocompromised being considered as non-responders. Should they not be part of the exclusion criteria since the sample size would be affected? Yes, I mean, if you've already thought about it beforehand at the design stage, you would maybe have not included them as eligible. But I'm just talking about in many situations, investigators often have not thought out perfectly who to not include. And so some people later on should not have been included. Okay. Or that's when these things come in. So that's what I'm saying. This is a little bit of a difficult, touchy situation to figure out. But so it depends on who the investigator thinks is likely to show a effect of the intervention, right? If they, for example, are not totally receptive to it. So yeah, maybe they got, let's say the drug is a drug, but their metabolism doesn't take on the drug, right? So it is not that the drug is not effective. It is that a person's metabolism doesn't absorb the drug for whatever reason. I don't know. I'm not a clinician, but that would be a non-responder. So it's something unique of a person that is different from other people that makes them not able to actually develop whatever is necessary to develop in order to then study the effect of it on the outcome of interest. Okay.
Okay. No
Any
more
other questions? No more questions. So then I would just thank everybody for their attention. I hope this has been an interesting set of lectures, and enjoy your evening.
Yeah. Thanks, Kant. Thanks. This is a wonderful lecture. We all enjoyed it. So I'm sure that we'll contact you for a few more lectures in future. Thank you.
Okay.
Thank you.
Thanks. Bye-bye.
Thank you, sir.
Thank you.
Thank you, sir.